my another vlog. For today's vlog, I will discuss about DNA replication. Why replicate DNA? DNA is the genetic material that defines every cell. Before a cell replicates and is divided into new daughter cells through either mitosis or meiosis, biomolecules and organelles must be copied to be distributed among the cells. DNA found within a nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. The process of DNA duplication is called DNA replication. DNA replication is the biological process of producing two identical replicas of DNA from one original DNA molecule. DNA replication occurs in all living organisms up still as the most essential part of biological inheritance. This is essential for cell division during growth and repair of damaged tissues, while it also ensures that each of the new cells receives its own copy of DNA. The cell possesses the distinctive property of division, which makes replication of DNA essential. Replication follows several steps that involve multiple proteins called replication enzymes and RNA. In eukaryotic cells, such as animal cells and plant cells, DNA replication occurs in the S phase of interphase during the cell cycle. Deoxyribonucleic acid, commonly known as DNA, is a nucleic acid that has three main components: a deoxyribose sugar, a phosphate, and a nitrogenous base. Since DNA contains the genetic material for an organism, it is important that it be copied when a cell divides into daughter cells. DNA is a type of molecule known as nucleic acid. It consists of 5 carbons, deoxyribose sugar, and phosphate, and a nitrogenous base. Double-stranded DNA consists of 2 spiral nucleic acid chains that are twisted into a double helix shape. This twisting allows DNA to be more compact. In order to fit within nucleus, DNA is packed into tightly coiled structure called chromatin. Chromatin consists of one chromosome during cell division. Preparation for replication. Replication for formation. Before DNA can be replicated, the double-stranded molecule must be unshipped into 2 single strands. DNA has 4 bases called adenine, thymine, cytosine, and guanine that form pairs between the 2 strands. Adenine only pairs with thymine and cytosine only bends with guanine. In order to unwind DNA, this interaction between base pairs must be broken. This is performed by enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into a Y-shape known as replication fork. This area will be template for replication to begin. DNA is directional in both strands, signified by 5 and 3 ends. This notation signifies which side group is attached to the DNA backbone. The 5 end has a phosphate group attached, while the 3 end has a hydroxyl group attached. This directionality is important for replication as it only progresses in the 5 to 3 direction. However, the replication fork is bidirectional. One strand is oriented in the 3 to 5 direction, while the other is oriented 5 to 3. The 2 sides are therefore replicated with 2 different processes to accommodate the directional difference. Primer binding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the three ends of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primase. Elongation. Enzymes known as DNA polymerases are responsible for creating the new strand by a process called elongation. There are 5 different known types of DNA polymerases in bacteria and human cells. In bacteria such as E. coli, polymerase III is the main replication enzyme, while polymerase first, second, fourth, and fifth are responsible for error checking and repair. DNA polymerase III binds to the strand at the site of the primers and begins adding new base pairs complementary to the strand during replication. In eukaryotic cells, polymerases alpha, delta, and epsilon are the primary polymerases involved in DNA replication. Because replication proceeds in the 5 to 3 direction on the leading strand, the newly formed strand is conscious. The leading strands begin replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase that adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous. The newly created fragments are disjoined. Termination. Once both of continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced with appropriate bases. Another exonuclease propagates the newly formed DNA to check, remove, and replace any errors. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single undefined strand. The ends of the linear DNA represent a problem as DNA polymerase can only add nucleotides in the 5 to 3 direction. The end of the parent strand consists of repeated DNA sequence called telomerase. Telomerase acts proactive hubs at the end of chromosomes to prevent nearby chromosomes from fusing. A special type of DNA polymerase enzyme called telomerase catalyzes and synthesizes of telomerase sequence of the ends of DNA. Once completed, the parent strand and its complementary DNA strand coils into familiar double helix shape. In the end, replication produces 2 DNA molecules, each with 1 strand from parent molecule and 1 new strand. Replication enzymes. DNA replication would not occur without enzymes that catalyze various steps in the process. Enzymes that participate in the eukaryotic DNA helicase. DNA helicase unwinds and separates double-strand DNA as it moves along the DNA. It forms the replication fork by breaking hydrogen bonds between nucleotide pairs in DNA. DNA primase. DNA primase is a type of RNA polymerase that generates RNA primers. Primers are short RNA molecules that act as templates for the starting point of DNA replication. DNA polymerases synthesize new DNA molecule by adding nucleotides to leading and leading DNA strands. DNA gyrase. DNA gyrase unwinds and rewinds DNA strands to prevent the DNA from becoming tangled or supercoiled. Exonucleases. Exonucleases group of enzymes that remove nucleotide bases from the end of DNA chain. DNA ligase. DNA ligase joins DNA fragments together by forming phosphodiester bonds between nucleotides.